everyone, my name is Julia. It's so good to see you. I hope you all are staying healthy and safe. A little bit about my background before I get started. I have research experience in insect behavior, but today we're going to talk about bacteriophage. What are bacteriophage? What do they do and why are they important? Bacteriophage literally means bacteria eater. There are viruses that go in and target pathogenic or bad bacteria. There's good bacteria and bad bacteria? Actually, yeah, not all bacteria are bad, and that's important to know. Actually, there's bacteria on our skin right now, and there's bacteria in our intestines, which helps keep us healthy and helps, keep us functioning. But pathogenic bacteria or the bad bacteria, when they get inside, it's not a good thing. A couple of ways that pathogenic bacteria can enter our bodies are through contaminated water, eating contaminated food, or maybe getting an insect bite from an infected insect, or having a cut or scrape that got infected with something. So if we get sick with a bacterial infection, how do we treat it? Well, antibiotics are typically the first route and the most popular line of defense. But what happens when that pathogenic bacteria or that bad bacteria decides to hang out for a while, even though you're trying to get better by using antibiotics? That's called antibiotic resistance, and that's where bacteriophage come in. When antibiotic resistance has developed, bacteriophage are brought in and target that pathogenic bacteria and destroy them. So how do these bacteriophage go in and do that? And what are they made of to be able to do that? To explain this, we're going to make Milo the Myloviridae. Milo is a bacteriophage that has a very unique morphology or set of physical characteristics that makes him him. So let's assign different colors for different body parts for Milo to make it easy to remember what the different parts are and what they do. So the first structure is the head. And the head is really, really important because it contains DNA specific to the phage. So I'm going to use pink for Milo's DNA, which is a unique set of chromosomes that are only Milo's. Oops, get that on there. You can spell the word DNA with your Play-Doh, or you can just make like a twisty snake-looking thing like I did. So that's really important because that genetic material of Milo's is going to be injected into the bacterial cell. So attached to Milo's head is something called the tail sheath. And the tail sheath is a tube of proteins that Milo's going to use to transport the DNA down into that bacterial cell. The next structure is the base plate. I'm going to use blue for the base plate. The base plate is important because this is what the tail fibers or the legs of the bacteriophage are attached to. And these structures help Milo attach to the cell and get ready to inject his DNA. Now, the morphology or physical characteristics of tail fibers or the legs can also be specific to the phage. In the case of Milo, he's going to have tail fibers or legs that kind of resemble daddy long legs. But there are some phage that you can see online that have a different morphology or different physical characteristic. They have a long tube-like midsection, and then they have tiny little short legs like centipede style. That long tube or column that's attached to the head of the phage helps classify them as Cyphoviridae. Cypho is Greek for the word tube, and that's where that name comes from, because their tail sheath resembles a tube. So there's Milo. There's his tail fibers, his legs. To review, head contains Milo's DNA or chromosomal information unique to Milo. Tail sheath is a tube of proteins that's going to transport that DNA down into the base plate, which helps those tail fibers attach and help that DNA get into the bacterial cell. I hope you enjoyed this session with me. I had a lot of fun, And I hope your bacteriophage looks better than mine. See you next time.